you for answering a few questions about your work at MPI for the Auckland Beekeepers Club. It was nice to chat earlier and to discover that MPI's chief scientist loves bees as much as we do. While researching this, I was pleasantly surprised by how much MPI is doing for bees and apiculture in New Zealand, and I thoroughly recommend checking out their website. So, Richard, you're MPI's biosecurity senior scientist. Does that make you the government expert on bees?
I'll just say first too, thanks Ken for the invitation to talk today, and I really like that question as the first question, because whilst I had this wonderful privilege of working on honeybee health as a scientist, and as you say, I'm a senior scientist based at the Animal Health Lab, the MPI Animal Health Laboratory in Upper Hutt. There are a lot of others at MPI who have expertise that relates to honeybees, or that there are others that are specifically working on it. And I can think of some of my colleagues. So I work on a bunch of research projects with Hayley Praggett, who's a senior advisor in bee biosecurity. But there's also Ching Hai Fan. He works at the Plant Health and Environment Laboratory. That's MPI, another laboratory MPI has in Auckland. And Fan is an expert on mites. So he often looks at honeybees, and particularly if someone suspects there's an exotic disease on them. I have other colleagues like Ben Pitty. He's an epidemiologist, and he runs the exotic disease surveillance program with support from Assure Quality. So there's a lot of others at MPI. I'm really only mentioning biosecurity there, but of course we've got people in market access, and there's the entire branch of New Zealand Food Safety at MPI as well. So yes, in terms of honeybees, I work on them 100% of my time, and I really enjoy it, and particularly that chance to do something that's hopefully protecting the primary industries of New Zealand, and also for recreational beekeepers.
Most importantly, do you keep bees?
I do keep bees, Ken. I've been when I first started this role, it was about 6 years ago, and I didn't have a background in bees, and I've had saturation for the last 6 years. So I caved in a couple of years in and got my own hives, and I really love it. I tell you what, I personally love about beekeeping, some of the things that I really enjoy is the smell of the smoker, the smell of the hives, the smell of propolis, the smell of the wax, the honey. It's the smells for me. I feel like I could just shut my eyes. And we have one colony here on our residential property, and one of my favorite things is to go outside at night, and I'm kind of saying good night to the bees, but that smell at the moment as they're drying off the honey, and they're all at one side of the entrance, circulating there, and it's just such a wonderful smell. So I think I can be quite a bore, and for many who don't keep bees, just going on and on about them, but I'm pretty sure there's a lot of kindred spirits at the bee club up there in Auckland. Yeah.
Excellent. I find that most people don't get bored when you talk about bees all the time.
You're right. Yeah. Yep. Most people, you're dead right, actually. Yeah.
So how did you go from studying Adeli's alopecia to bees?
You've done your research, Ken, because I've worked on lots of different species, and you mentioned the Adelie penguins, and I used to work as a virologist, just specifically on viruses, and we would work on any species. So I worked as a medical laboratory scientist for about 10 years. I worked with the Crown Research Institute, ESR, and so we worked on all sorts of things, and animals, and lots of human work around viruses as well. So a lot of my work in the past when I worked there was focused on applying a technology, which is genomics sequencing technology that everyone seems to know about these days, what with COVID. But I saw an opportunity come up for a role at MPI that was working with honeybees. And I probably was a little bit frightened of them, but I've always wanted, because I did my training down at Otago University, I always wanted to stay in New Zealand and use my skills as a scientist for the betterment of the country. And I like getting outside a lot. And I have to say I was very intrigued by beekeeping. I think it seems like an interesting lifestyle for commercial operators. So that's what personally drew me to want to come across to this role. So I still use all the skills I have in laboratory background around diagnostics and genomics, but it's just been so enriching for me to get out and talk to beekeepers and just have that direct connection to what's happening with the science you're trying to provide. So if you work in the medical realm and you're a scientist, there's a person in between you and the patient called a medical doctor. But yeah, as I say, I was really interested in being able to have that close integration or contact with beekeepers. So yeah, and I've just found that to be really enjoyable. Yep.
Thank you. So I've been reading quite a bit about the bee pathogen program. What is that, and how does that? What are you trying to achieve with that?
So the bee pathogen program, it has finished now. It was a 3-year project that was funded by, there's an operational research program within MPI. So that allows us to do specialized projects from time to time that obviously will fit into support our operations. So what that project was based upon, and it ran from 2016 through to 2019, it was 3 years. And it was based upon the USDA, the US Department of Agriculture, and the University of Maryland run the Bee Informed Partnership in the United States. And there are other types of this research done around the world. And what they do and what we did was follow very closely in New Zealand, it was 60 beekeepers. We would go to their apiary that they nominated to be studied. We would do that in spring, and then in autumn, and spring, autumn, and then spring. And when we visited, we would take a sample of live bees, we would take a sample of bees preserved in ethanol, and we would look in there to see what viruses those bees had, how much of those viruses the bees had, how many varroa were in there, how much varroa. And then we were supported by a very talented and experienced group of inspectors who'd done various roles and other things. So they came on board to help us in different regions of New Zealand, and they would interview the beekeeper each time they visited to get an understanding of what their management practices were, and collected a lot of information about things like floral sources and various just management practices, and so what varroicides they're using, etc. So we have this very large data set, and we are still working through the analysis on that data set, but we recently published a paper which just described the general architecture of the project and some descriptive epidemiology about what we found. So, what viruses we found and how often we found them.
Thank you. That sounds like a great sort of database for us for the future. So the api well-being, is that following on from the bee pathogen program?
I mean, in a way it does, because the same, many of the same people are involved in that from within MPI, except that the api well-being project, we have been, it has a number of different pieces of work inside of it. So one of those is to build a lot more extension materials, and by extension materials we mean things that we can provide to beekeepers to help raise their awareness about biosecurity. And also we're working on looking at American foul brood, the genomics of it, so what genetic strains are present throughout New Zealand. And we've been really well supported with the collection of samples by the National Pest Management Agency for American foul brood. We're also doing some more retrospective testing of samples that we collected during the bee pathogen program for viruses that we weren't able to look for at the time we did the project. And we have also set up an architecture for people in the future who might want to access the collection that we amassed, because we'd be well over 130,000 individual honeybees frozen at -80 degrees Celsius. And of course, there's all this information attached to the bees. So we've sought permission from the participants, the beekeepers who are involved in this, that we would in the future be able to share that with other researchers in New Zealand, so that if they were going out to look for, to do a project, we wouldn't be collecting samples twice. So that is also an aim of the AP Wellbeing Project, which is coming into its final stages. So we're just paused to deliver on some of the things that we said we would do during the project, which I've mentioned just there.
Mm-hmm. And how significant is genomic sequencing of Penibacillus in the fight against AFB?
And I'm going to focus on the sort of word, how significant is it? I guess one is always at pains to point out that, the most significant thing that beekeepers are doing at the moment in New Zealand is, of course, their legal obligation to inspect their hives and destroy hives if they find that Penibacillus larvae, American foul brood, is present, and to notify the National Pest Management Agency for American foul brood. So that's the highly significant part. But when we started the AP Wellbeing Project 3 years ago, genomic sequencing from the world I came from working on lots of human diseases and that was being done a lot, but it was really starting to be done on AFB. And there was a very good study in Sweden that was done by Eva Forsgren and co, where they looked at an island in Sweden that didn't have AFB, and they looked at the mainland of Sweden, and they were able to track the movements of people who had basically where AFB had come from when someone took some secondhand beekeeping gear from the mainland across to the Scotland island. So the purpose of our project that we're doing is not to create a tracking tool for looking at AFB. All the samples we've collected have been anonymized. What we are doing with our project is just looking for to understand the genetic types that are present in New Zealand. And we do know that there are lots of changes within AFB that could affect virulence. So, how dangerous it might be. But there are two broad types that people might hear about. There's the ERIC 1 type and the ERIC 2 type. And around the world, both are discovered. There are a number of other ERIC types which are much more rare. So yeah, this is one thing that we hope we can sort of shine some light on in a larger way as we've collected well over 150 apiaries right across New Zealand that have had American foul brood. And as I say, we've been supported by the AFB agency. They've collected samples for us. They're given to us, and we don't collect anybody's personal information. We're just interested in the general region of New Zealand where they came from. So for you guys, we would call that the Auckland region. And you can, the other regions might be, say, Mid Canterbury, North Canterbury, Southland, Coromandel. And we're just going to go through, look at the whole genome sequences, much like is done with COVID at the moment, and just trying to understand what the spectrum of genotypes are in New Zealand. So I think it's important information that's needed, but yeah, there's many tools, and I think with dealing with AFB, that's what the AFB agency is doing. Yep.
Thank you. The annual colony loss survey shows some interesting trends. We seem to consistently hover around 10% winter losses. Has the reported reason changed much over the years?
So I guess it's always important to point out that our colony loss rates are comparatively low compared to some that are reported in other jurisdictions overseas. So I think that is a good thing. But queen problems has tended to be reasonably consistent. but I know we have seen every year slight increases in the number of colonies that beekeepers attribute Varroa to having caused a loss. So perhaps that's a concern, but we've got, really great work being done by Manaki Whenua. So that's Pike Storman Brown, who works there. He's the principal economist that does the survey, and it's just great to be one of I think there's at least 35 countries around the world that perform these types of surveys annually. So it's an excellent piece of work that MPI commissions Manaki Whenua, Thank You Research, to do every year. And yeah, I think that the concern still is around Varroa and watching that attribution creep up every year.
And beekeepers are asked to report the reason for the colony losses. How accurate do you think that their diagnosis of the loss is?
I think that there's complexity in that question because it probably somewhat depends on what the agent is. So things like Nosema ceranae, they might be quite hard for particularly a novice beekeeper, but even experienced folks to see, because it's a microorganism, and it often results just in colony death with no preceding symptoms. So that is a tricky one. Yeah, having said that, beekeepers know their bees, and when they turn up to a colony and see an end point, if it is currently overwhelmed by wasps, or it's got vast amounts of parasitic mite syndrome in it that's been caused by Varroa, one thinks that they're going to know what they're doing there. It's just pointing out perhaps it's not, the colony loss survey doesn't have laboratory tests behind it, or we don't have inspectors verifying things in the field. But a real positive with the colony loss survey is that such high participation rates from New Zealand beekeepers. Massive participation rates compared to countries overseas, which see sort of single digits. We can see over 30% of beekeepers in New Zealand making those contributions, and particularly because it's been carried over a number of years. The accuracy, if you like to call it, of beekeepers is one thing, but that should be fairly consistent across the years. So if there's trends, that's a real effect that we'd be seeing. So it's looking at those trends over time, but yeah, beekeepers, I think, do know their bees, and there are many things that you could say. Poisonings, like a poisoning event's pretty easy to spot as well. Yeah, that I think that it's really valuable, valid data.
Excellent. Besides the bee biosecurity section of the MPI website, where else can we learn more about bee diseases in New Zealand?
I think many in the club and yourself, Ken, would support this. I think a really good port of call is peers, fellow beekeepers, particularly those with experience, lots of experience, those who, bee clubs, for example, such as the Auckland Bee Club. I think they do a great job. And for new people looking to enter the field, there's the industry training organisations. Around American Foul Brood, there's the AFB Pest Management Agency, and they have lots of great resources online. And then if you're wanting to read more widely, we've got the MPI Bee Biosecurity webpage, which is a good touch point for, as you've mentioned. So pretty much if you just Google Bee Biosecurity and MPI, there's our work that we're doing to provide materials for beekeepers. But you can look around overseas. There are some good resources in other countries. Looking across the ditch at Australia, they have some really good bee biosecurity work and resources being made if you're interested in looking at what in other countries been writing. Yeah.
Cool. So what do you think is the next big threat to honeybees in New Zealand?
That's a tricky one, and I guess within my role working in biosecurity New Zealand, I will view things through a biosecurity perspective, and that's what I feel I could probably speak to. So the things that we have concern about that are present overseas but not in New Zealand, it's agents like European foul foul brood, Melissococcus plutonius, which is a bacteria that causes brood disease, brood death, and you can look up pictures of that. There's small hive beetle that's causes problems for hives all around the world. Australia has it. Australia has European foul brood as well. That's something that we're concerned about. Tracheal mites present in many other countries, so Acaripus woodi, that can cause really substantial colony deaths when it first arrives in a country. And there's a number of other agents that we don't have in New Zealand that I think we're very keen to keep out, and therefore raising biosecurity awareness amongst beekeepers is just it's a central role, really. That's what we do. So yeah.
And the similar question here, what do you think the biggest threat to beekeeping in New Zealand, or apiculture in New Zealand?
And once again, it's a complex system, beekeeping, because I understand there are lots of economic pressures and market sort of aspects, and it's not really my field of expertise. So from what we understand from beekeepers is that Varroa is still really a real challenge to manage, even 20 years on from it first arriving in New Zealand. And then there is the for those who find their hives affected, there's American foul brood. Those are challenging things. Yeah.
Thank you. And if a hobbyist beekeeper has a hive with a suspected disease, can they send a sample to MPI? If so, how would they do it?
So this is very straightforward. We have an 0800 number that you can call and talk to someone we've got. And that number is a 24 hours a day number. We have investigators who have pages they walk around with, and those pages will go off, directing them to respond to a call. So we would ask that beekeepers call 0800 80 9966, so that's 0800 80 99 66, and they're going to get directed to one of the people at MPI who's going to know all of the capability we have, and we'll take an interest in trying to find out what's going on. But we really encourage people to do that if they have if they see something and they consider it could be an exotic organism or pest that's causing the problem, or if they have large scale hive losses as well. If you lose 80% of your hives, for someone with a larger number of hives, that's really concerning, and we would ask that they call that phone number as well. That's our central point for gathering and getting ready to detect exotic disease.
And what was the last book that you read on bees or beekeeping?
I love reading books on beekeeping. I actually happen to have a copy here. This one is by Sue Hubble, A Book of Bees. Oh, it's blurring it out. There you can see. Sue Hubble, I think she's passed away. She wrote another great book as well. I've just really enjoyed reading that. She's in, let me recall, one of the southern states that slips my mind in the US, but it's got that relaxed, considered approach that beekeepers, I think, kind of live with as they just slow down when you go in a hive. When you're having a look in a hive, I feel like that when I'm reading the book. At the same time, I was reading. I just had gone to actually read Langstroth's book, and it's pretty slow. It's pretty tedious. There's a lot of kind of allegorical almost writing in there. So I go back to Sue Hubble's book to brighten my day up a bit when it's been getting a bit slow. But I thought I must read some of these seminal texts in Langstroth's book. I think it's probably just an important thing, a bit like Moby Dick. You've got to have a crack at it. So yeah, that's me at the moment.
And one final question. Isn't it a bit too windy down in the capital for bees?
I love it. I am from Otago, so I lived down there for the first 30 years of my life, and I've lived up here for about 15 years. Because we live in the hut, I don't think we get as much wind as Wellington. Having said that, I know that there's some very talented beekeepers along there at the Wellington Bee Club who seem to get vast amounts of honey. So, you know, you got to say it. You can't beat Wellington on a good day. Maybe that's what the bees are up to, is they just, wait for the gap and get out there and pull that Pahudakawa honey in.
Breeding stronger flyers.
Yeah. We did have, myself and Hayley had spent some time on the Chatham Islands, for our research work. And it's very interesting to look at the challenges the beekeepers have there, but they're doing a great job. And they, it can be quite windy there. They said there's sometimes, where you see the bee come out of the hive and then zip. But, they make it back, and obviously they have some great days too, because they have some very interesting honeys over there. So, yeah.
That's my next holiday down there, because they don't have any disease down there. And they've got the original bees that were introduced to New Zealand, haven't they? The black bees, British black bees.
Yeah. I think that they've got some curious sort of lineages there. They don't have varroa, so they're very keen to keep that out, as you can imagine. So yeah, a really cool place to visit, and just fascinating place.
Yeah, with this COVID though, there seems to be quite a waiting list to get into there now.
Yeah.
The only overseas place you can get to.
Yep.
Well, thank you for your time, Richard, from Auckland Beekeepers Club, and hopefully we'll see you at one of our field days soon.
Thanks, Ken. Yeah, it's a real privilege to be in the role I am as a scientist, and just be trying to do something in earnest, as a public servant to help protect what we've got here, because it's still a special thing, and it's an important part of our primary industries as well. So yeah, thank you for the opportunity to talk to you today, and thank you to the club for the invitation.
Excellent. Thank you very much, Richard.
Okay, thanks, Ken, and
Cheers.
catch you later. Okay, see you.